Hi guys, so next topic we are going to discuss is about the antibodies and the immunoglobulins. So what are the antibodies? So antibodies are the substances which are formed in the serum and in the tissue fluids in response to the antigen and react with that antigen specifically. So basically these are formed in the serum. So there are a number of immunoglobulin. So what are immunoglobulins? They are the molecules that are produced by the plasma cells in response to an immunogen. Immunoglobulins are the glycoprotein. Each immunoglobulin, it is composed of the 2 light chain and 2 heavy chain. These are the 2 light chains and these are the heavy chains. So the light chain has the molecular weight of about 25,000 while the H chain, the molecular weight is about 50,000. Now the light chain, these are attached to the heavy chains. The light chains, these are attached to the heavy chain by disulfide bond. This is the disulfide bond. And the two heavy chains, these are also connected to each other with the help of the disulfide bond. Or the disulfide bridge. So immunoglobulin isotype, that is immunoglobulin G, M, A, D, E, it is determined by the type of Ig, immunoglobulin heavy chain present. These are very important classes. So the immunoglobulin class IgG, in the heavy chain the gamma is present, IgA alpha, IgM mu, IgD delta and IgE epsilon. Fine. So the light chains, these are similar in all the classes of immunoglobulins, either kappa and lambda, but in the kappa it is being more common, important. In the light chain, in all the classes of immunoglobulin, the light chain, either the kappa and the lambda, these are in the similar classes, but kappa it is being more common. So antigen combining site is at the amino terminus and it is composed of both the light chain and the heavy chains. Both the light and the heavy chain consist of the two units, the constant and the variable. See, this is the variable region and this is the constant region. So both the light as well as the heavy chain, they both contain, this is the light chain and this is the heavy chain, they both contain the variable region as well as the constant region. Fine. So the heavy chain, one variable VH and 3 or 4 constant unit. So in the heavy chain there is one variable and 3 or 4 constant region. While in the light chain there are one variable and one constant unit. In light chain there is one variable and there is one constant region. So the variable region is further subdivided into hypervariable region and framework region. This variable region, this one, it is further divided into hypervariable region and framework sites. Fine. And the hypervariable regions at the amino terminal are involved in the formation of antigen antibinding sites. Important. So the light chain has the 3 hypervariable region and the H chain has the 4. So the hypervariable regions, these are also called as hotspots. Fine. So the sites on the hypervariable region which make the actual contact with the epitopes are called as complementary determining region, CDRs. Fine. These are known as complementarity determining regions. So the infinite range of the antibody specificity of the immunoglobulin, it basically depends on the variability of the amino acid sequences at the variable unit of heavy and light chains. So the Fc fragment, it is composed of a carboxy terminal. It determines the biological activity such as the complement fixation. The CH2 of the IgG and CH4 of the IgM, they bind to C1q portion of C1. Fine. Now coming on to the heavy and light chains. First of all, let's discuss about the heavy chains. So there are 5 isotopes and the classes of the antibodies. The isotope variation or the class of antibody, it is due to the variation in the amino acid sequence in the constant region of the heavy chain. In this constant region of the heavy chain, this region, constant region of the heavy chain, because there are the 5 potential variation in the amino acid sequence in the constant region, there are 5 classes of antibodies. What are the antibody? G, M, A, D, E. So these H chains, the H chain differences, these are called as isotypes, and these are designated by the Greek letter gamma, alpha and mu, epsilon and delta. So coming on to the light chain, all the 5 classes or the isotypes have the same amino acid sequence on the light chain. The 2 light chain isotope designated as kappa and lambda. So it means in the heavy chains you have the chain differences which is called as isotypes, and these are designated as the Greek letters like the gamma, alpha, mu, epsilon and delta, while in the light chain, the 2 light chain having the isotypes, that is kappa and lambda. And the important point is, in the light chain, all the 5 classes or the isotypes have the same amino acid sequence. Fine, but in this there is a variation in the sequence. Now, what is the constant region? So the amino acid sequence in the constant region of both the heavy and the light chain, it is similar for all the antibodies molecule within each class. Important. Variable region. So in this, the amino acid sequence of the variable region of both the heavy and the light chain varies with the antigen towards which the particular antibody is directed. So the portion of the variable region which has a highly variable amino acid sequence, it is known as hypervariable region, as we already have discussed. And the hypervariable region, it is also known as hotspot number 1, and the hypervariable region at the amino terminal are involved in the formation of antigen and antibody sites. So the hypervariable region of the H and L chains, these are associated with the form of the epitope binding region which is known as antibody idiotype. Antibody idiotype. So what is this? This is a part of an antigen which binds to the antibody. It is known as epitope. So sites on the variable region which make the actual contact with the epitopes, these sites are known as the complementary determining regions, which is CDRS and paratopes. Important. The infinite range of the antibody specificity of the immunoglobulin, it basically depends upon the variability of the amino acid sequence, as we have already discussed this line. Coming on to the hinge point. So the point at which the heavy and the light chain join, this is known as hinge point. This point can be digested by the papain digestion. Important. Fine. So let's discuss some of the points, some of the important points or the points to be remembered. The basic immunoglobulin molecule, it is composed of 2 heavy chains and 2 light chains which are connected by the disulfide bond. So the light chains, these are consist of the 2K chains, that is the 2 kappa chains and the 2 lambda chains, while the heavy chain, it belongs to the 5 classes AGA, G, M, D, E, which is known as your epsilon, delta, alpha, gamma and your mu. Fine. Now it is a class of a heavy chain that determines the class of the immunoglobulin. See guys, it is a class of an H chain which determines the class of an immunoglobulin. So it is also important to know that the constant portion of the heavy chain, that is not variable region, that determines the class of the antibody. So this is something which is very important for your MCQ point of view. So all the immunoglobulins are heat stable except the IgE. It is only the immunoglobulin which is the heat labile. Only immunoglobulin which is secreted by the seromucinous gland, it is IgA. Immunoglobulin present in the milk are IgA and IgG. The immunoglobulin to fix the complement via the alternate path are IgA, IgD, IgG. Immunoglobulin to fix complement via classical pathway IgG and IgM. The immunoglobulin with the maximum serum concentration IgG. So the antibody with the least serum concentration is IgE. Important. Immunoglobulin with the maximum molecular weight is IgM. Immunoglobulin with the maximum sediment coefficient is IgM. Immunoglobulin with the least carbohydrate is IgG. Immunoglobulin with the maximum carbohydrate is IgE. Immunoglobulin with the maximum half-life is IgG. And immunoglobulin with the minimum half-life is IgE. These are very important points. Starting with the immunoglobulins, various types of immunoglobulins, starting with the IgG. So it is the major serum immunoglobulin which constitute 80% of the molecular weight of the half-life. That is the only immunoglobulin that only transport or through placenta, and it is also responsible for complement fixation and it protects the body fluids. These are the 3 important points. So starting with this, it is the major serum immunoglobulin which constitute 80%. It is distributed equally between the intravascular and the extravascular compartments. It contains less carbohydrate and than the other immunoglobulins. It has a maximum half-life. So it binds to the microorganism and enhances the phagocytosis. So considered as a general purpose antibody, it protects against those infectious agents which are active in the blood and the tissues. So it is predominant antibody in the secondary antibody response. 4 subclasses of IgG are there. Ig1, it is found in greatest amount, while Ig4 it is found in the least amount. So IgG1, it is found in the greater amount, while IgG4 it is found in the least or in the smaller amount. So Ig2, it is directed against the polysaccharide antigen, so it is important in defense against the encapsulated bacteria. IgG, it is the only maternal immunoglobulin that is normally transported through the placenta and provides the natural passive immunity in the newborn. This one. Right? So it is the only maternal immunoglobulin that is normally transported across the placenta and provides the passive immunity in the newborn. So there are 4 subclasses of the IgG. As I've already told you, IgG, it is found in the greatest amount, while IgG4 it is found in the least amount. So IgG1 constitutes 65%, IgG2 23%, G3 8% and G4 4%. Next is IgM. So IgM, it is the earliest immunoglobulin that It is the earliest immunoglobulin class to be synthesized by the fetus and beginning at 20th week of the age. So as it is not transported across the placenta, the presence of the IgM in the fetus or the newborn indicate the intrauterine infection, because it is not transported across the placenta. It is the IgG, it is the only immunoglobulin which is transported across the placenta and provide the passive immunity to the newborn. So it is IgM, it is normally circulates as pentamer. It is the oldest immunoglobulin class. So if a question comes like which of the following is the oldest immunoglobulin class, belongs to oldest immunoglobulin class, so your answer should be IgM. So it is a predominant antibody in the primary antibody response. It is short-lived, disappearing early than IgG. Hence, its demonstration in the serum indicates recent infection. So treatment with 2-mercaptoethanol selectively destroyed the IgM without affecting the IgG. This is a simple method for the differential estimation of IgG and IgM. So in this, the isohemagglutins group, blood groups anti-A and anti-B, these are usually IgM. Being largely confined to the intravascular space, the IgM it is believed to be responsible for the protection against the blood invasion by the microorganism. So the monomeric IgM, it is the major antibody receptor on the surface of the B lymphocyte for the antigen recognition. So IgM, it is an important component of the immune complexes in an autoimmune diseases, that is, example, the IgM antibodies against the IgG in rheumatoid arthritis. So IgM antibodies these against the IgG in the rheumatoid arthritis. So after IgG, IgM, let's discuss IgA. So IgA, it is the second most abundant antibody after IgG. It is the major immunoglobulin in the colostrum, in the saliva, present in the tears, respiratory and GIT secretions. IgA occurs in the two forms, serum IgA, secretory IgA. Serum IgA, it is the monomer found in the serum, while the secretory IgA, it is the dimer. Serum is a monomer, secretory is a dimer. Two monomer units are joined together by a glycopeptide J chain and contains the glycine-rich polypeptide as a secretory component, important, which protect the IgA from denaturation by bacterial proteases. This is the J chain. So the two monomers, these are the two monomers, these are unite together with the help of a glycopeptide which is a J chain. Fine. So SIAG, that is the secretory IgA, and the J chain, these are synthesized by the plasma cells. Secretory component, it is produced by the mucosal and the glandular epithelial cells. There are two subclasses of IgA that has been described, IgA1 and IgA2. IgA1, it is predominant serum IgA, and IgA2, it is predominantly secretory IgA. So this is IgA1 and this is IgA2. Right? So after IgA, IgG, IgM, let's discuss the immunoglobulin IgE, which is known as a reagent antibody. So IgE resembles the IgG structurally. Properties are, it is heat labile. As I've already told you that IgE, it is the only immunoglobulin which is the heat labile, while all the other immunoglobulins, these are heat stable. So affinity for the surface of the tissue cell, mast cells, and the homocytotropic, these are the species specific. It cannot be demonstrated by the conventional serological reactions such as the precipitation or the complement fixation. So it is detected by the RAS, which is a radioallergosorbent test ELISA, and passive agglutination. So chiefly produced in the lining of the respiratory and the intestinal tract, and it is responsible for anaphylactic type of hypersensitivity. Elevated in the atopic condition like type 1 hypersensitivity, parasitic infection and anaphylaxis. In this you can see there is an increase or elevated level of the IgE. Coming on to the immune responses. So immune response, the antibody response to an initial antigenic stimulus which differs qualitatively and quantitatively from the response to subsequent stimuli with the same antigen. So former it is called primary response and later it is called the secondary response. So in this there are two types of responses, the primary response and the secondary response, but first it is known as primary response and later it is known as secondary response. Coming on to the primary response, the primary immune response refers to the first encounter of the immune system with an antigen, the antibody that is elicited first in the entirety of the IgM type. So IgM antibody titer rises to the peak level, then decline almost as fast as it develops. Firstly, the IgM titer, it increases to the peak level at the starting, but when it is fastly developed, the level of the IgM it gets decline. So in a few days IgG appears. After the IgM, after few days the IgG appears, and this again the IgG titer gradually increases at the beginning, then decline gradually. An important outcome of the primary antigenic challenge is the production of the memory cell or the primed cell. Coming on to the secondary response. So a secondary response refers to the re-encounter of the same antigen against which the memory cell, that are the T and the B cells, have developed during the primary exposure. See, it is the later response and this is the primary is the former response. That is why a secondary response, it is recounter of the same antigen against which the memory cells, the T cells and the B cells, have developed during the primary response. So as the primed cells, these are already present in the antibody response, it is very fast and there is no lag phase as compared to primary response. There is a brief production of IgM, but major antibody is IgG. But in this, the IgM antibody was formed and it increases at the beginning, but at the later level when it gets fastly developed, the IgM it gets declined, and later the IgG antibody it get developed. But in the secondary response there is a brief production of IgM, but there is a major antibody is IgG, which is produced in the larger amount for the longer duration. Now last is the difference between the primary and the secondary response. So the primary response, it is slow, the secondary response is fast, the primary response is sluggish, the secondary response is powerful, primary response is short-lived, secondary is prolonged, antibody persists for long. And which antibody? Mainly the IgG. Primary response has the long lag phase, while the secondary response has the short and the negligible lag phase. Antibodies appear in the short time. And in the primary response, predominantly IgM, and in the secondary IgG. Antibody, it is more specific in primary response, while antibody is less specific in the secondary response. Antibody is less avid in the primary response, while it is more avid in the secondary response. In primary response, it is a qualitative response, while quantitative response is in the secondary response. And at the last, I am discussing about the role of the different immunoglobulins classes. So IgG, it protects the body fluids. IgA, it protects the body surfaces. IgM, It protects the blood stream and IgE, it mediates the reagenic hypersensitivity. Fine. So guys, this is all about the antibodies and the immunoglobulins. Please go through it once and try to make the notes. Thank you.